Hello students. So we are going to learn about bacterial flagella today. So many prokaryotic organisms are mobile by swimming motility, and this function is because of a structure called as flagella. The flagellum functions by rotation of its flagella to push or pull the cell through a liquid medium. Bacterial flagella are long, thin appendages, free at one end and attached to the cell at the other end. Bacterial flagella are so thin, about 15 to 20 nanometers in diameter, depending on the species, that a single flagellum can be seen under light microscope only after being stained with special scales that will increase their diameter. Flagella show different arrangements on the surface of a bacterial cell. In case of polar flagellation, the bacteria are attached at one or both ends of the cell. Occasionally, tufts of flagella may arise at one end of the cell, which arrangement is known as propellers flagella. If such tufts arise from both ends of the cell, then the arrangement is known as amphitrypous flagella. In peritripous flagella, flagella are inserted at many locations around the cell surface. Coming to the structure of the flagella, flagella are basically not a straight but actually a helical structure. The filament of the flagellum is composed of many copies of the protein subunit, which is known as flagellum. Flagellum proteins are highly conserved in the amino acid sequence in species of bacteria. The flagellum consists of several components and moves by rotation, similar to the motor boat. The base of the flagellum is structurally different from the filament and is wider at the base of the filament. The portion is known as the hook. Hook is made up of a single type of a protein and acts to connect the filament to the basal body. The basal body is basically the motor portion of the flagellum. The motor or the basal body is anchored within the cytoplasmic membrane, and it consists of a central rod that passes through a series of rings. In case of gram-negative bacteria, the outermost ring is known as the L ring, that is anchored within the lipopolysaccharide layer. The second ring is the P ring, that is anchored in the peptidoglycan layer, while the inner two fused rings are MS ring and the C rings, which are embedded in the inner membrane and the cytoplasm. In case of gram-positive bacteria that lack the outer membrane, only the inner pair of rings are present, that is MS and the C rings, that is anchored in the inner membrane and cytoplasm. Surrounding the inner rings are anchored in the cytoplasmic membrane a series of proteins which are known as mock proteins. Another final set of proteins which are known as flea proteins function as motor switch that reverse the direction of rotation of the flagellum in response to intracellular signals. So flagellum is basically like a tiny rotary motor, and it has two main components, the rotor and the stator. The rotary component of the flagellum motor consists of a central rod surrounded by the four rings L, P, C, and MS rings. Collectively, this structure makes up the basal body. The stator component consists of the mock proteins that surround the basal body and function to generate torque or force that will allow rotation of the flagellum. The energy required for the rotation of the flagellum comes from the proton moving force. Proton movement across the cytoplasmic membrane through the mock complex drives rotation of the flagellum, and about 1000 protons are translocated per rotation of the flagellum. There are several genes that are required for flagellar synthesis. The process is most widely studied in bacteria such as E. coli and Salmonella typhi. where it has been identified that more than 50 genes are linked with bacterial motility. These genes have several different functions, including encoding structural proteins of the flagellum and the motor apparatus, export of the flagellar proteins through the cytoplasmic membrane to the outside of the cell, and regulation of many biochemical events surrounding the synthesis of new flagellum. A flagellar filament basically grows from its tip. During formation of the flagellum, first the MS ring is synthesized and inserted into the cytoplasmic membrane. Then other anchoring proteins are synthesized along with the hook before the filament forms. The flagellum protein molecules are synthesized in the cytosol and they pass through the narrow 3 nanometer channel inside the filament and they are added at the end to form the mature flagellum. At the end of the growing flagellum, protein cap exists, which can assist flagellum molecules that have diffused through the channel to organize at the flagellum terminal to form new filaments. Approximately 20,000 flagellum protein subunits are needed to make one filament. Broken flagella can still rotate and can be repaired with new flagellum units. Coming to the moment of the flagella, two different types of moments are being identified in flagella. The first type is identified in bacteria having Peripheral flagella. So the forward moment is imparted by counterclockwise rotation of the bundle. Clockwise rotation of the dumbbell causes a tumbling effect. And again, the flagella bundle again, and they move in the counterclockwise direction to lead in a new direction. In case of bacteria that have polar flagella, they again move by counterclockwise rotation of the flagellum in the forward direction. They can change the direction by rotating the flagella in the reverse direction, that is clockwise direction. Flagella can rotate about up to 300 revolutions per second and open the cell frequently at up to 60 cell lengths per second. The slow motion of polarly and lymphotropistically flagellated organism differs from those of peripherally flagellated organism. Peripherally flagellated organism typically move in a straight line in a slow and a deliberate fashion, while polarly flagellated organisms, on the other hand, move rapidly by spinning around their flagella, and it seems like a dashing movement from place to place. The different behavior of the flagellum from polar and peripheral flagellum, peripheral organism, including differences in the reversibility of the flagellum. Thank you.